From Isaiah 43, a verse that kind of sets the stage for what I'm going to be talking about during Sabbath School. It says, "Thus saith the Lord that created you, O Jacob, and that formed you, O Israel." So this is the God, the Creator God speaking. "I have redeemed you. I have called you by your name, and you are mine." So God claims us as His. He tells us He has redeemed us, and the reason He can redeem us is because He created us. Doesn't matter whether we believe in the biblical account of creation or not. Doesn't matter. It's fundamental to understanding salvation. And we're not going to talk about that this morning, but I just want to set the stage for my remarks. And where do we learn about God's creatorship? Do we learn it by studying the world around us? Or do we learn it from this? So the Bible is fundamental to our understanding of both nature and salvation. And I just want to emphasize that first so that my remarks won't be taken out of context, because we're going to talk about the scientific evidence. Scientific evidence supports the story in the Bible. It does not establish the story in the Bible, it supports it. So it's very important to understand that relationship because it's fundamental to everything I'm going to say. Now, I'm going to tell you some amazing things this morning. I hope you will enjoy and appreciate them. But bear in mind that this is supporting and enhancing and enlarging the view that we have of God that comes from the Bible. Why do I believe in creation? I believe it because of this. Even though I'm a scientist that spent many years doing science, I believe in the creation account in the Bible because it's in the Bible, not because of the demonstrations that I see in science. Nonetheless, we do find a lot of support in biology. And we're going to talk about two different areas. First of these is molecular biology and origin of complexity. I don't want anybody to worry about those big words because we're going to get around them. See, I have a typo on my very first slide. I'm sorry. Computers, proteins, and the human brain. What does this have to do with creation? Well, in 1999, IBM, International Business Machines, started out a project called Blue Gene. Now, it's a play on words, of course, but the idea is IBM is Big Blue, and they were going to study genes. They were going to build a computer so big that it could figure out how to make a protein with a specific structure. Now, how big was that computer? They planned to build a computer with 1 million processors inside it. 1 million. Now, they didn't quite make that in the first iteration. I think they had a couple 100,000, but they have gotten there since then. They wanted to predict how a protein would look after it was made, because the idea was man wants to control nature. We want to have rulership over nature. And if we could learn how to make a protein with a specific structure, then we could design proteins that would do things we want them to do. So that was the goal of building this big supercomputer. That would solve all our problems because, of course, if we could design a protein, then we could think of something to do with it, and that would fix everything that ails us. That was the goal. This is the biggest computer in the world. It's called Sequoia. It has 1,600,000 computers inside it. And you can see them going on in both directions. It does 20 petaflops, which is far more than anybody 20 years ago thought would be possible. Interestingly, the very first iteration of Blue Genes, they dedicated for a whole year solid processing trying to solve the protein problem. After a whole year of trying to solve the problem of how to predict the structure of a protein, suddenly you never heard any more about it. Is that because they were successful? No. And now they have computers that are much bigger. This one is 5 times bigger than the original one and 20 times more powerful. And they don't waste any time trying to solve the protein problem. So then they turn to other problems like the brain. Maybe we can generate enough information to understand how the human brain works. And of course, if we understand that, we can solve all the problems of mankind. The fastest supercomputer was thought to be able to cover about 1% of the activities of the human cortex. That's what they were saying. We're within 1/99th of being able to generate a computer big enough to do what a human brain does. Then, just this week, there came out a new article in New Scientist. They have discovered that the human brain has 86 billion neurons, each of which is like a computer. Each of which itself functions as a computer. It says in the article, they call it a PC. So we have in our brain 86 billion computers. That was hoped some years ago that we could be able to solve the problem of making a robot that could think. We wanted to make something artificial with artificial intelligence that could think. That was our goal. We thought, I remember reading a few months ago an article about how close we were to doing this. Well, you know, close only counts in horseshoes and hand grenades. Close doesn't count when you suddenly discover that there are 86 billion neurons in the human brain, each of which is acting like its own computer. So we are far, far away from the possibility of making an organism which has the same capabilities as the human brain. Very, very far removed. The world's most powerful computer working for an entire year was not capable of predicting the shape of a single protein molecule, something that evolutionists believe accidentally formed with a function that made life possible. Accidentally. We know that it can't possibly happen accidentally. Yet otherwise intelligent beings believe that functional proteins arose by some kind of cosmic accident. Isn't that interesting? I'm not saying these people aren't smart, but when it comes to understanding origins, they are far away from where the data lead us. Let's talk about developmental biology. Everybody in this room started out as a single cell. Single cell. You were at one time one cell. How do you make a single cell into a human being? How does that happen? What kind of information and understanding has to be built into that system in order to make it become you, a single cell? Have you ever wondered how that could happen? Let's take a look at a salamander. Salamanders are amphibians and they lay eggs, so we can look at their egg and watch it develop. And let's do that. Here's a single salamander egg. And what we're going to see is the egg divides, and we have two cells. And from that point on, the divisions are asymmetric because we are symmetric about an axis, but we're not spherically symmetric. So right away, that egg starts going through a procedure which is governed by regulations that are built into the system. And I want you to watch. I'll try to point out some of it. Some of it you'll see for yourself as it divides. There's the first division. This is all speeded up in time, of course. Now there are 4 cells. Now watch the next division. It establishes the polarity of the egg, and one end is going to be the head and the other end is going to be the tail. So already at this very early stage, it knows what's going to be what in the final product. Now we have a morula. Cells are dividing. Now we're going to initiate the formation of the germinal pore. The gastrula, and gastrulation is going to occur. Now, how do all these cells know they're supposed to go inside? Look at them. They're moving. They're being orchestrated by something beyond them. These cells aren't just suddenly falling down into this hole. They know that they're supposed to go there, and they're going there. This hole will become establish the differentiation of the different tissues of the body. And there you can see the pore. That will be the gut. Inside of that will be the gut. That's your mouth. And now we're going to form the nervous system. Here you see the neural plate forming, and then the folding to form the nerula. That's the spinal cord along the back, and then the brain up at the top. People, who's telling those cells what to do? Now something really amazing is going to happen. You're going to see a bunch of cells that are not part of a tissue running around on the surface, going places. Do you watch carefully? There, you see them? They're all going places. They're going to become something specific, and they have to go somewhere else to do it. You there? They're moving. They're going in order to seed a new part of the organism. And now I'm going to share with you something I heard on National Public Radio last week. So I was listening to them talking about the sexual systems and how sex development takes place. A part of the embryo, of the human embryo, is a bag called the allantoin. And the allantoin is where the embryo, the developing embryo, puts all its waste products. It dumps the garbage out there. Well, out of this allantoin, there are a series of about 40 cells that are differentiated, and they are going to march single file up into the embryo, and they're going to go to a place where the gonads develop. So if you're a woman, it will be the ovaries, and the male, it will be the testes, but the process is the same. So these 40 cells march single file, holding hands along a corridor. They march up there to where they get where the gonads are, and then they split off, one going to the right, one going to the left, one going to the right, one going to the left, until all of those 40 cells are distributed to the 2 gonads. If you're a male, they embed themselves in the wall of the testes, formative testes, and if you're a female, they go into the ovary and they start replicating, and they make all the eggs that will become your children while you're still in the uterus. So your children are established while you're still in the uterus. Who controls all this stuff that's going on? Who tells those cells to hold hands and march to the north? Who tells them where to go? Who tells them to split off one by one and go to opposite sides? Amazing. I think we should watch the rest of this probably, just because it's only begun. Here you can begin to see the body axis, the head over here, the tail over here, the eye forming, the limb buds. Okay, now it's time to get out of that bag. You see the bag there. It generates enzymes, and those enzymes go out and digest that, so it falls away. Here you can see the blood starting to move. Right there, here's the heart. Circulation of the blood. You can see the cells moving there. And it's beginning to look like a salamander, or at least like a newt. Here's the heart. Here's the gills. Here's the blood circulating in the gills. Heart beating. And now it's time for him to break out of the egg sac. He can't break out of that. It's much too tough. Just then, in the process of the development, an enzyme is produced which digests away the material the egg sac is made of. And you'll see him be able to get out. There's blood circulating in the gills. Okay, watch now. He's going to digest the end of this with that enzyme. Notice it's not his efforts that get him out. There goes. There you are. He's born. Now remember, we started with a single undifferentiated cell, and we made an organism as complex as any organism there is in just a few hours. All that programming had to work flawlessly in order for this to happen. Any mistakes and he wouldn't have been born. Taking this over to the bigger picture, all complexity of modern organisms is present at the beginning of the fossil record. When we first see fossils, and that's what we're here looking at in Tucson this week, when we first find fossils in the rocks, those fossils already have all of this complexity that you saw there that made this egg turn into an organism. Because we know that for many reasons, but one is because those organisms started out as an egg also, and they differentiated into whatever they became. No proposed mechanism for origin of complexity can stand careful scrutiny. If you try to explain to me how that egg became a salamander, you're going to stop real quickly, because you just do not have the machinery. You do not have the tools to make that out of nothing. No example of a preserved sequence of development of complex function or even a single functional protein is known. Nobody can give me a step-by-step explanation for how we got a protein from a non-proteinous material. Okay, those are fundamental to being able to talk about evolution. If you don't have those, how can you speak to me about evolution? All right, let's talk about genetics. That's Dr. Phillips' domain. But we'll talk about it a little bit. The Human Genome Project. We kind of thought we understood us pretty well in the year 2000 when we completed the sequencing of the human genome. At least we said we did. We found something astonishing. We expected that there would be 100,000 or more genes in the human genome because we make 100,000 different proteins, at least in our bodies, probably many more than that. And yet, when we actually found the genes on the DNA, there were only about 20,000 of them. That's far short of what we expected. We thought humans would have more than anything else. It turns out 20,000 is kind of a round number for any organism. So people began to wonder, why is it that we have 100,000 proteins if we only have 20,000 genes? They began to look to find out how it is that we can get more than one protein from a single gene. And we're going to see how that happens in a minute. But remember, in order to make this work, you have to have an order of complexity more control. You have to control how you put these genes together to make different proteins. And we now know that, for example, a protein called the DSCAM protein, which is fundamental in making the nervous system develop, we know that it can make 19,000 different proteins from one gene. How many? 19,000 proteins from one gene. Now, why would the body need 19,000 proteins? You can take a bird egg, isolate it from its mother, raise it in an incubator, hatch out a chick, and then raise that chick up, and that bird that's never seen its mother will sing the song that its mother sang. How does that happen? Or you have a little child. You bring the child home from the hospital and you smile at that child when it's not crying. And the child looks up at you and it smiles back. Did you ever think about that? What's involved in recognizing a smile on somebody else's face and turning that into a movement of muscles in the face of the baby? What is involved in that? The complexity of that. It's all wired in. And what about, let's say, a giraffe? A giraffe, I speak of giraffes because they have a traumatic birth. They fall several feet to the ground. That's their rude awakening. The first thing they do is get up on their feet and walk. How do they know how to walk? Where does that coordination come from? You think it's easy? Ask a human baby as they're trying to learn to walk. Now, fortunately for us, God didn't make humans so they could pop out and walk off. We'd be in trouble from the get-go. But God at least gives us an adjustment period of a year or so to get used to having this little thing around before it goes walking off to get into trouble. So that's divinely inspired, I'm sure. But other organisms, they're born and they can walk. The pattern to know how to walk is built in. Many, many other things are built into the brain as it develops. The brain is not just a mass of undifferentiated tissue. It is a mass of carefully programmed information that is laid down first so that when we are born, we can do something. And how does that all happen? Well, the DSCAM gene is part of it because as this cell divides, and it's going to become this neuron over here, and this cell divides and it's become this neuron over here. Those cells recognize and talk to each other, and the DSCAM proteins are different in those two cells. Each cell that is produced generates a different DSCAM protein for the next cell, so that the whole thing is programmed out by the differentiation of this gene. Wow. Who knew? And tell me, how much regulation does it take? How much information does it take to program into a gene the ability to pattern out the path of neurons in the brain so that it functions? How does that all work? Do you have any ideas? Well, apparently proteins like the D-scan protein and others are laying out these patterns of neurons so that our brain actually does function when we're born. That's amazing beyond belief. Not only do you need the genes, which we have no clue how to produce those without a designer. Not only do you have to have the genes, you have to have these systems of higher regulation that specify how those genes are going to be allocated. Another example, your ear. Along the course of the cochlea are neurons that are spaced in such a way that they can respond to sounds of different frequencies at different places along the neuron. How do those cells know what frequency they're supposed to respond to? There's a single protein, one protein, that produces 980 different proteins that are distributed along the cochlea in order of frequency, so that in this cell down here it responds to a C sharp, and this one recognizes some other frequency next to that, and so on, all the way along the cochlea. 980 different isoforms. How do they know which one to make? Because they're all made from one gene. Well, let's watch how that happens. Here you can see a DNA molecule right here, and here's a DNA-dependent RNA polymerase making an RNA molecule. Now, the RNA molecule will make the protein. And there are parts of that RNA molecule right here called introns, which have to be cut out and thrown away. And here you can see a special machine called a spliceosome. It has about 30 different proteins in it that comes along, and it grabs the two ends of this intron, and it brings them together, and it joins together the ends and throws it away. And then the two exons, the part that's going to exit the nucleus, are fused, spliced together. So here you see it happening. There the end of one is released and it's joined to the end of the other to make the RNA molecule right here. This is the intron. It's going to be thrown away and degraded. Recycled. And there goes the spliceosome. So this is a whole transcript, DNA transcript in the form of RNA. It has 3 introns in it. These 3 introns will be spliced out and thrown away, seemingly thrown away. Some of them have functions as well. And then this is the exon. This is going to go out into the cytoplasm and make the protein. Now, there are 3 pieces of RNA that are joined together here. This is a very simple example of what these bigger RNAs will do. You could take this piece and join it with this piece and leave this one off. You could take this piece and join it with this piece, or you could take this piece and join it with that piece. So you can make 3 different proteins from this particular strand of RNA. Do you see how that works? Well, if you have 50 different pieces that you can join together to make the RNA, you can see that you can make a lot of different proteins with that. 19,000 in the most extreme example I know of. But again, I want to remind you, it takes a higher level of control to be able to deal with a gene like that needs to be told which one to make now. And that's all programmed in. So now evolutionists, when they saw that only 2% of the DNA was genes, they started saying, well, look, 98% of the DNA in the human is junk. That's where the evolution's taking place. So you have all this evolution going on out there, and eventually something new comes out, and then that becomes becomes a new gene, and we make a new protein. But only 2% of the DNA is making proteins, and we thought that's all the DNA did. That was our illusion about DNA at that time. So 98% is junk. But some people knew that that wasn't the case because that 98% stayed the same from one organism to another. some of it. And so they started a project called the ENCODE project. I don't know what the acronym stands for, but it involved 440 scientists and 32 labs around the world. They studied to see what the 98% of the DNA that evolutionists were calling junk, if that really had any function. They developed tests for activity, and they found out at least 80% of the junk DNA was doing something in the cell. At least 80%. The protein coding genes, that is the 2% of the whole DNA that we thought was the real stuff, turns out that's the same in everybody. It's the same in a monkey, it's the same in a mouse, it's the same in a worm. We all have a complement of genes we have to be able to function as a living being. Our coding DNA, that is the genes, is 50% similar to a worm, 95% similar to a mouse, 98% similar to a chimpanzee. Does that mean we're related to a worm, a mouse, or chimpanzee? No. It means we have to have basic fundamental things that are similar across the spectrum of organisms. With the 2% of the DNA being similar, it must be the 98% of the DNA that is the important part. Why? Because we're not a mouse, we're not a worm, we're not a chimpanzee, even though we have a lot of the same DNA. So it must be the 98% that makes us what we are. Let's look at a machine shop analog to help understand this. This is a machine shop. A machine shop is full of drawers that have tools. If you have a good machine shop, it's got a lot of drawers, and they have tools neatly arranged in the drawers. Here's an example of a drawer in a machine shop. These are all tools that are used in the making of things. But we need two more things in a machine shop to make it do anything. The tools in the tool drawers will not make a thing. Is that right? They're very important, and you can't make anything without them. But you can't make anything with them unless you have some other things. So they're like the genes. They are the genes, the 2% of the DNA that we thought was everything. That's like the pieces in a machine shop. The two other things you need are a blueprint, a plan. How am I going to end up with the thing I'm trying to make? And what else do you need? Need a machinist. Somebody who will operate the machine, use the tools, and produce the product on the blueprint. So, going back to the cell, the DNA are the tools, the drawers in the tool shop, the tools that are in the drawers. The blueprint and the director of the activity has to be outside of that 2%. And that's what the rest of the DNA is doing. And nobody thought about that. Early on, we thought, well, we understand genes, we understand organisms, we understand life. Now we're saying, we don't know a thing. We're starting from scratch. The ENCODE project has liberated us forever from the possibility that we are the result of some cosmic accident. Clearly, we have a master planner who has encoded our DNA, in our DNA, the operating blueprints, the directions for proceeding, and the machinations to accomplish the goals. That's the end of the idea that we evolved from nothing. You can't go beyond that. There is no way around this wall. So the ENCODE project has shown us the futility of trying to think about evolution being an accident. And evolutionists know this. And so even though these are top scientists around the world, they have fought those scientists tooth and nail to try to discredit their work. Because they know that if this is true, this is true. But that's not all. Let's just take a look at this. This is a molecule of DNA. The molecule of DNA in one nucleus of one cell, the molecules of DNA are 6 feet long. If you take all the DNA out of that cell and line them up, it's 6 feet long. It's compressed into a nucleus that is 10 millionths of a meter across. In order to make that happen, we have to use these protein molecules here called histones. The histones wrap the DNA around themselves and make a thick thread. And by the way, notice those histones have tails. We're going to be talking about those in a minute. And then those threads coil up to make a supercoil. And then that supercoil coils again to make a supercoil. And then that supercoil coils again to make a supercoil. And that's how our DNA becomes a chromosome. And that's how it fits inside the nucleus of the cell. Now, when it's coiled up like that, here you can see the cell dividing. Those are the chromosomes that you just saw made of single DNA molecules. When it's coiled up like that, the DNA can't be read, it can't be copied, and therefore the cell can't do anything. In order to read that DNA, we have to uncoil it, uncoil it, and then we have to open it up in such a way that the DNA strand is visible again. That brings us to the subject of epigenetics. The dogma was that genes were everything. The environment had little to do with development. The idea of Lamarck was that a giraffe got a longer neck because he reached higher in the trees. And these ideas were scoffed at by a generation of geneticists. But during the past 2 decades, we've gradually learned that something else is involved that was not genetic, in the sense it isn't involving the genes themselves. The studies that were done, they took mice and they exposed them to a pleasant odor that they had never smelled before. And then when the mice were exposed to the odor, they would shock them on their feet, and the mice would go running off. After they had done this a while, the mice learned that when they smelled that odor, they better run. Even mice are smart enough to figure that out. Well, mice are smart enough to figure that out. Sometimes humans have trouble. So when they were shocked, they would run off when they were exposed to this pleasant odor they'd never been exposed to before. So that was an interesting experiment. They trained them like that, then they bred them together. And they took the offspring, removed them from the parents, raised them up, and then they exposed them to the same odor that mice had never been exposed to before. And what do you suppose happened? They ran. They ran. Why? Somehow, something that those parents had done had produced a signal in the egg and sperm that were passed to the offspring that caused the offspring to recognize and respond to something that mice had never recognized or responded to before in the history of the world. It was passed to the offspring not by genes or genetic information, but by a whole new sophisticated system that we didn't even know existed, that we refer to now as epigenesis or epigenetics. It has profound implications for us, because it means that our behaviors as adults can be passed to our offspring and can affect their lives. Here was the description of that in Nature magazine, the most sophisticated science magazine in the world. This is the article about that study that I just mentioned. And its title was Epigenetics, the Sins of the Father. Now, where does that phrase come from? Where in the Bible? From Exodus chapter 20. The second commandment, the sins of the father will be visited upon the children of the third and fourth generations of them that hate me, and showing mercy unto thousands of them that love me and keep my commandments. There is hope, by the way. There is hope. But that's what they found with the mice. The first generation, they responded. The second generation, they responded. The third generation, they responded. By the fourth generation, they lost the ability to respond to that stimulus. So just as the Bible said, and just as I had been teaching my students for years, there is a consequence of our behavior in our offspring. The Bible said so. I didn't know the reason. Now I understand a little bit more about the reason. I'll show you a little bit about that in a second. But it's true. And it's right. And our hope is that God says, showing mercy to thousands of them that love me and keep my commandments. Here's how it works. Remember how the DNA coiled around those protein molecules called histones? Here is the histone. Here is the DNA coiled around it. These are those tails I pointed out to you, sticking off from those protein molecules. These are specific amino acids. Let's see, that would be, what's K? K is histidine, R is serine, is arginine, S is serine, and T is threonine. I think that's right. Anyway, these are 4 amino acids in the tail. Here's 1 in this tail. There are 2 in this tail, and there are 3 in this tail. Each of those amino acids can be acetylated with either acetone or methane, methanol, or a phosphate group, an acetyl group, methyl, methylphosphate, phosphate, ubiquinone, acetyl group, ubiquinone. That's a total of 15 different moieties that can be put on those amino acids. Each of those constitutes a change in the behavior of this whole nucleosome here. So that's 15 different places. Well, those 15 to 2 to the 15th power is about, what, 64,000? Is that right? Rebecca? Does that sound right? 2 to the 15th power? I think it's 64,000. There are 64,000 different ways that this particular nucleosome can be affected by these little signals put on there. 32,000. Thank you, Susan. 32,000 different ways that these can be affected by those signals. This is one nucleosome core. There are thousands of nucleosome cores all the way along the DNA molecule that are all affected by this process in different ways. So it's like a huge concert piano or a huge concert organ being played, and which keys you push depends on which kinds of noise you get out of the system. All of that regulation mechanism here is involved in epigenetics. It doesn't change the DNA. The DNA remains the same, but it changes when and if that DNA will be copied to make a protein that will affect the cell somehow. So the cell has to have an additional, sophisticated, higher level of regulation. Somebody is in there telling it which ones of these amino acids need to be methylated or acetylated or whatever, elated. And so there has to be a regulation of that. Somebody has to know which ones to do. How sophisticated is it? I can transfer information about an odor that the mouse has never been exposed to before, and a response to that odor. I can transfer that through the patterning of these things on the outside of the genes. That will then go into the next generation. That mechanism has to be controlled, so there must be another regulatory mechanism above this. Evolution is completely bereft of an explanation for this level of sophistication. We can't do anything with that.
You're good, I'll wave you down.
Okay. Isn't this fun? Then the other shoe fell. Not that there was another shoe to fall, but if there was, it fell. When we discovered that there are genes in every organism which do not exist in organisms that are supposed to, by evolution, supposed to have given rise to them. So there are genes in the human that are not present in the chimpanzee. Not a few. As many as 500 genes that humans have that chimpanzees don't have. Now think about what that means. Well, maybe they don't do very much. Maybe they're just genes and they're non-functional. One of those genes that humans have that chimpanzees don't is responsible for the large brain that we have. Interesting, isn't it? Honeybees, which look just like other kinds of bees, honeybees have a whole series of genes that are not found in any of the other bee families that are responsible for them making honey. God said, here, you can make honey, and we'll enjoy that. You may get stung in the process, but that'll be something for humans to enjoy. Those genes did not evolve because they're not found in any of the things that we think might have given rise to bees. They're not found in any of the things that could have come from bees. They're found in honeybees and nobody else. Those are called orphan genes. Orphan genes are the biggest indictment. The simplest to understand indictment of evolution is the theory of origins that there is. Because there are not just a few. There are at least 500 genes that we have in humans that are not present in chimpanzees. If we had come from the chimpanzees over a period of a couple million years, or from a common ancestor with the chimpanzees over a period of a couple million years, then there's an awful lot of change going on that produces important genes that are responsible for us being humans and not chimpanzees. And we don't have a mechanism for that. We can't explain how even one gene that has a function can come into existence from a non-gene. How can we explain how 500 in humans, at least 500 in humans, came into existence? Where did they come from? Evolutionists would expect to find similar genes in related organisms. Orphan genes defy evolutionary explanations to this point. So what do we do? How do we deal with this data? Can we still be an evolutionist in the face of so much conflicting data? Evolution by random processes and natural selection lacks explanatory power for the existence of even one functional protein. We can't explain if IBM's supercomputer cannot figure out how to make one protein running for a whole year with 1 million processors. How can we explain the existence of a protein in 1 million years, or 100 million years, or 1 billion years? It doesn't matter the number you pick. No conceivable naturalistic mechanism can explain the existence of the human brain. How are you going to explain it? Did it just gradually develop from other organisms? Well, no, those other organisms have that same complexity. So where does it start? There is no organism born with a brain that is not pre-programmed. Because if it were true that the brain wasn't pre-programmed, what would the thing do when it was born? The genetic discoveries of the past 20 years defy explanation within the evolutionary paradigm. There isn't anybody that can stand here and say, oh, I've got an explanation for that. I'd love to hear it. It would be amusing. Evolution stands stripped of its explanatory power in the light of modern science. And I'm going to stand on that. Thank you. Oh, yeah. All right. Let's have prayer. Gracious Father, we thank you for the revelation of your character through the world of nature. We understand that this revelation is understood through the revelation of your word. That you are the Creator God, and because you're the Creator God, you are the Redeemer. We ask that you will help us to use this information to glorify your name. In Jesus, amen.